Welcome to The Explainer. Today, we are taking a journey from the code inside our very own cells all the way to the birth of new worlds. We're going to explore 4 recent scientific breakthroughs that are completely rewriting what we thought was possible.
I mean, just look at these questions. What if the answer to all of them was yes? Well, today we're looking at breakthroughs that are making that happen. Let's start with a revolution that's taking place right inside the human body. All right, our first stop is a truly historic medical advance that was announced back in December 2023. It offers brand new hope for a devastating genetic disorder. Okay, so this disease is caused by just a single tiny mutation, but its effects are anything but small. Those sickle-shaped cells, they get stuck in blood vessels, right? And this creates blockages that lead to these episodes of excruciating pain. People have described it as feeling like sharp glass moving through your veins, and it causes severe organ damage. These events have a name: vaso-occlusive crises. And just to put this into perspective, in the United States alone, about 100,000 people are affected by sickle cell disease. For them, this isn't just some abstract medical condition, it is a painful, daily reality. Now, this is what's so incredible. The FDA approved 2 totally different, groundbreaking gene therapies on the exact same day to fight this one disease. On the left, you've got Casgevy. This is the first ever approved therapy that uses CRISPR technology, the famous molecular scissors that can precisely edit DNA. It tweaks a patient's own stem cells to get them to produce fetal hemoglobin, which is a type that doesn't sickle. Then on the right, you have Lyfgenia. It takes a totally different route. It uses a harmless virus as kind of like a delivery truck to carry a new functional hemoglobin gene into the cells. Just 2 brilliant paths to the same life-changing destination. And as Dr. Verdon from the FDA points out here, this is way more than just a new drug. It is a fundamental shift in how we think about disease. We're not just managing symptoms anymore. We're using a patient's own modified cells as a one-time treatment to fix the problem at its genetic source. This is the leap from just treating something to actually curing it. And the results? Honestly, they are staggering. Just look at these numbers. In the clinical trials, an incredible 93.5% of patients treated with Casgevy were completely free from those terrible pain crises for at least a year. Lyfgenia was right behind at 88%. For people who have lived their entire lives in constant pain, this isn't just an improvement. It is a whole new life. So you see, this power to edit our genetic code is all built on our understanding of the human genome. And that map just got a massive update. We're going from fixing a single gene to finally completing the instruction book for an entire human chromosome, one that's been a total mystery for decades. Imagine trying to put together a puzzle where half the pieces are just identical solid blue. That's kind of the challenge scientists were facing with the Y chromosome. Its structure is so incredibly repetitive that for decades, our technology just couldn't figure out how to piece it together, leaving more than half of it as a total blank spot on our genetic map. But then, in August of 2023, using brand new sequencing technology, this group called the telomere-to-telomere consortium finally filled in all those gaps. And as the lead researcher Adam Philippi said, what they found wasn't just chaos. It was this surprisingly elegant, almost beautiful quilt-like pattern of DNA. So you're probably asking, okay, but why does this actually matter? Well, having this complete map is like having the full architectural blueprint of a building for the very first time. Researchers can now study these medically important genes in a way they never could before. They can see the full structure of the region that's critical for sperm production. They can analyze how these weird DNA palindromes, where the genetic code reads the same forwards and backwards like the word radar, can lead to infertility. And they've even discovered that the number of copies of a key sperm gene can vary from 10 to 40 between different people. It's a game changer for understanding fertility. Okay, so we've gone from editing our genetic blueprint to finally completing it. Now, let's zoom way out from the human scale to the planetary scale and look at a breakthrough that's trying to rewrite the chemical blueprint of our atmosphere by tackling one of the biggest challenges we face: carbon emissions. So, the big dream here is basically a form of industrial alchemy. What if we could capture the carbon dioxide pouring out of major polluters like steel plants or cement factories, and instead of just burying it, we use electricity to turn it into useful stuff, valuable chemicals like ethylene and ethanol that we use every day? And it turns out the secret ingredient that makes this whole thing possible is copper. It has this unique ability to do this conversion really well. But to make the process efficient enough to actually use on an industrial scale, scientists really needed to understand exactly what was happening at the atomic level. In a study from February 2024 revealed something totally counterintuitive. You'd think the best catalyst would just be made up of the most hyperactive atomic sites, right? But it turns out the ones that work the best actually have a mix of those superactive sites and less active reservoir sites that act like a support team. And this quote from researcher Christopher Hahn just illustrates it perfectly. It's not about the individual atoms doing all the work. It's about how the entire network functions as a cooperative team, creating this little supply chain that feeds the most active parts and makes the whole system way, way more efficient. It's all about teamwork. So for our final breakthrough, we're going to take a huge leap from our planet deep into the cosmos to watch a story unfold that, really, it mirrors the dawn of our very own solar system. Let me introduce you to SC Chameleontis. It's a young star that's basically a baby picture of our own sun from about 4.5 billion years ago. And it's surrounded by this huge disk of dust and gas, the exact same raw material that the Earth and all the other planets in our solar system were built from. Okay, now here is where things get really weird. Back in 2008, NASA's Spitzer telescope saw this strange neon signal coming from the star, which suggested the disk was getting blasted by UV radiation. But when the James Webb Telescope looked at it just 15 years later, that signal was gone. Now it looks like the disk is being hit by X-rays. So the big question is, how could it change so much, so fast? And this isn't some small academic difference. The type of radiation hitting that disk completely changes the rules for making planets. Think of it like this. You're trying to build a sandcastle. X-rays are like a really fast, destructive tide that comes in and just washes away all your sand, the planet-forming gas, really quickly. It gives you less time to build. And how much of a difference are we talking about? While computer models show that a disk bathed in that gentler UV radiation gives planets a full 1 million more years to form before all their raw material gets blown away into space. 1 million years. In the race against time for a planet to be born, that's a huge, huge advantage. So, what's the solution to this cosmic mystery? Well, scientists now think the star has this powerful, flickering wind. And most of the time, this wind actually blocks the UV light, letting only the X-rays through. It seems the Spitzer telescope just got incredibly lucky and happened to catch the system during a rare, quiet moment when the wind died down. And what that suggests is that the window for planet formation might actually flicker open and closed, a dynamic, pulsating process we have never actually seen before. So when you pull it all together, from our own genes to distant galaxies, these breakthroughs show that science is advancing on every single front, all at once. We're learning to rewrite our own biology, we're completing the human instruction manual, we're trying to re-engineer our planet's chemistry, and we're literally witnessing the birth of new worlds. And really, that's the key thing to take away from all this. These aren't just isolated events. They show an incredible acceleration in the pace of discovery, from the building blocks of our bodies to the formation of distant solar systems. We are constantly peeling back new layers of the universe and our place in it, and every answer just seems to lead to more fascinating questions. The future isn't just coming, it's arriving faster than ever.